Hello students, here we will learn diagnostic tests and treatment of diseases given in your NCERT book from term 2 syllabus. First, we will check all diagnostic tests. First test is Widal test. It is used to diagnose typhoid disease. Second is ELISA test, enzyme linked Immuno sorbent assay test. It is used to diagnose AIDS and it is based on the principle of antigen-antibody interaction, जिसमें पैथोजेंस को डिटेक्ट करने के लिए या तो हम प्रेजेंस ऑफ एंटीजेंस को चेक करते हैं या फिर एंटीबॉडीज़ को डिटेक्ट करते हैं जो हमारी बॉडी में पैथोजेंस के अगेंस्ट डेवलप होती हैं. इन्फेक्शन बाय पैथोजन कैन बी डिटेक्टेड by the presence of antigens or by detecting antibodies synthesized against pathogen in our body. Third test is PCR, polymerase chain reaction test, which is very important for early detection of diseases like AIDS. It is an accurate way to diagnose certain infectious diseases at early stages. HIV pathogen को detect करने के लिए and nowadays you are all aware that this test is also used to detect coronavirus. इसके अलावा PCR is also used to detect mutations in genes in suspected cancer patients. Let us now see the test to detect the cancer. Check the steps involved to detect mutated genes which are causing cancer. Number 1 step is single-stranded DNA or single-stranded RNA, radioactive molecule is taken and this is called as probe. Second step, these probes are allowed to hybridise to its complementary DNA in a clone of cells. Probes अपने complementary DNA strands के साथ hybrid DNA बनाएंगे. Next step is इस hybrid DNA को detect किया जाएगा by using autoradiography technique. Now check the results. हमें क्या दिखाई देगा फोटोग्राफिक फिल्म पर. द क्लोन हैविंग अ म्यूटेटेड जीन विल नॉट अपीयर ऑन द फोटोग्राफिक फिल्म. जिन क्लोन्स में म्यूटेटेड जीन्स हैं वो फोटोग्राफिक फिल्म पर अपीयर नहीं होंगे बिकॉज प्रोब विल नॉट हैव कॉम्प्लीमेंटरिटी विद द म्यूटेटेड जीन्स. म्यूटेटेड जीन्स के साथ कंबाइन करने के लिए प्रोब के पास उसका कॉम्प्लीमेंट्री बेस सीक्वेंस नहीं है, इसलिए म्यूटेटेड जीन फोटोग्राफिक फिल्म पर अपीयर नहीं होंगे. नाउ चेक अदर टेस्ट टू डिटेक्ट द कैंसर. फर्स्ट इज बायोप्सी. इन दिस अ पीस ऑफ सस्पेक्टेड टिश्यू कट इनटू थिन सेक्शंस एंड आफ्टर स्टेनिंग इसको एग्जामिन किया जाएगा अंडर माइक्रोस्कोप. डायग्राम में देख सकते हैं लिवर टिश्यू का पीस निकाला जा रहा है. दिस विल बी कट इनटू थिन सेक्शंस and then this will be stained and examined under the microscope to test the cancerous cells. This is called as biopsy. Second is blood and bone marrow test किए जाते हैं to check increased cell counts in case of leukemia or blood cancer. Third, in radiography, X-ray images are very useful to detect cancers of internal organs. Fourth test to detect cancer is computed tomography और CT scan technique. यहाँ भी एक्सरेज का यूज़ किया जाता है to generate a 3-dimensional image of the internals of an object. CT scan में 3-dimensional image ली जाती है internal organs की to detect the cancer. Fifth test is MRI or magnetic resonance imaging test. In this, strong magnetic fields and non-ionizing radiations are used. With this, we can accurately detect pathological and physiological changes in the living tissues. So, all these techniques are used to detect various diseases in the human beings. Now, next section is treatment of diseases given in your NCERT book. Number 1. एंटीबायोटिक्स का यूज़ किया जाता है टू ट्रीट मैनी डेडली डिजीजेस लाइक प्लेग, whooping cough also called as kali khansi. Then diphtheria and leprosy are also treated by using antibiotics. First antibiotic discovered by Alexander Fleming was penicillin. Number 2, vaccines and immunization programs have completely eradicated deadly disease like smallpox. Smallpox completely eradicate हो चुकी है by using vaccines. Some diseases like polio, diphtheria, pneumonia and tetanus have been controlled to large extent. And now we are also fighting with corona-19 disease and trying to come out of it by using vaccines. Drugs like antihistamine, adrenaline and steroids are used to reduce symptoms of allergy. Anti-retroviral drugs are given to patients suffering from AIDS, but they are only partially effective. To treat cancer, combination of radiotherapy and chemotherapy and surgery are used. In radiotherapy, tumor cells are killed by using radiations, but we have to take care of normal cells surrounding the tumors. In chemotherapy, certain chemotherapeutic drugs are given to kill cancerous cells, लेकिन इसके भी बहुत से साइड इफेक्ट्स हैं like hair loss, anemia, etc. Next, some biological response modifiers like alpha-interferon are given to cancer patients to activate their immune system, जो ट्यूमर सेल्स को डिस्ट्रॉय करने में हेल्प करते हैं. Come to next, morphine. This drug is very effective, sedative and painkiller and is used in patients who have undergone surgery. Streptokinase, a bioproduct obtained from bacterium Streptococcus, it is used as clot buster. ब्लड वेसल्स में क्लॉट्स को रिमूव करने के लिए स्ट्रेप्टोकाइनेज यूज किया जाता है फॉर द पेशेंट्स हु हैव अंडरगॉन मायोकार्डियल इन्फार्क्शन और हार्ट अटैक. Cyclosporin A, another biomolecule produced by fungus Trichoderma polysporum, it is used as immunosuppressive agent in organ transplant patients. Statins, produced by fungus Monoscus purpureus, used to lower blood cholesterol level. बेसिकली यह उन एंजाइम्स के एक्शन को कम्प्लीटली इनहिबिट कर देते हैं जो कोलेस्ट्रॉल सिंथेसिस में हेल्प करते हैं. कम टू नेक्स्ट, जेनेटिकली engineered insulin, which is used to treat diabetes. Earlier, this insulin was extracted from slaughtered cattle and pigs, but this insulin caused patients to develop allergy. Now, insulin is produced by recombinant DNA technology. Eli Lilly, an American company, prepared it in 1983 by using plasmid of bacteria E. coli. Next disease is ADA disease, adenosine deaminase deficiency disease. It is a hereditary disease caused by deletion of a gene and the enzyme adenosine deaminase is unable to form. This enzyme has crucial role for the immune system to work and it can be cured by number 1 bone marrow transplantation, number 2 enzyme replacement therapy जिसमें फंक्शनल एडीए दिया जाता है, लेकिन इन दोनों केसेस में कंप्लीट क्योर डिजीज का पॉसिबल नहीं है. थर्ड मेथड इज जीन थेरेपी. फर्स्ट जीन थेरेपी वॉज डन ऑन 4-year-old गर्ल इन 1990 जिसमें फंक्शनल एडीए ssDNA को इंट्रोड्यूस किया गया इनटू आइसोलेटेड लिम्फोसाइट्स ऑफ द पेशेंट्स. इस जीन को इंट्रोड्यूस करने के लिए रेट्रोवायरल वेक्टर का इस्तेमाल किया गया. आफ्टर इंट्रोड्यूसिंग दिस फंक्शनल जीन into lymphocytes of blood of the patient, it is then returned back to the patient's body. As lymphocytes are immortal, a periodic infusion of these genetically engineered cells are required. लेकिन अगर जीन को एम्ब्रियोनिक स्टेज पर ही इंट्रोड्यूस किया जाए, then it could be the permanent cure. So that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.